Good morning. I'm Caroline Troopgill. Welcome to this news briefing from the 252nd National Meeting and Exposition of the American Chemical Society in Philadelphia. We're joined today by Dr. Marya Lieberman from the University of Notre Dame and Sarah Bliese from Hamline University. They will be talking to us about their work on paper-based device that can identify falsified and degraded medicine. So, Dr. Lieberman?
Well, thank you. I guess maybe I'd like to start by introducing people to the problem that we're trying to solve, and really this is our central problem. These are medications that are very effective against malaria, and so they're really important from a health standpoint in large parts of the developing world. And one of these packets of medicine is a falsified product. It doesn't contain the active ingredients, it's just starch. And so the problem is that this kind of medicine is really difficult to detect in the low resource settings where it tends to appear. And that's the problem that we're trying to solve. And really this touches on issues of basic fairness and justice. If people are paying for medicine, they deserve to get medicine. And so this is the project that we've been working on.
So this issue affects people from every level. So it affects the patients that are supposed to be receiving these medications and then not getting the treatments that they're supposed to be. It affects the caregivers, such as the pharmacists and doctors that are prescribing these medications, because it starts to make them question their confidence in the medical system. And then it affects the buyers that buy it directly from the manufacturers, because they start to question the quality of the investment that they're making into this pharmaceutical. The solution that we've developed is a paper analytical device. It's a simple test card about the size of an index card that Marya can display here.
So I brought some. This is a little foil pack that we have, and we can put about 20 of these cards in the pack. And a single card can be used to test many different kinds of medications. So this card is designed to test antibiotics and tuberculosis medications, but it also works for some other drugs. So, for example, for some diabetes medications and heart medications. And that kind of drug for chronic conditions is becoming a lot more important in the developing world. And so the test cards are inexpensive and they're very easy to use. So it gives people who have a question about a medicine something that they can use to do a quick screening test to determine if the medicine is falsified or not.
And then based off of these screening tests, we can follow it up using high-performance liquid chromatography, which is the gold standard for testing pharmaceuticals after these samples have been sent to Notre Dame. But asking Notre Dame to take on the task of analyzing every pharmaceutical that's collected is rather hefty. And so Notre Dame has partnered with other institutions around the nation to assist in this analysis, which is where my university, Hamline, came in. We analyze ciprofloxacin, and while we have these samples, we're able to look for different components that affect the quality of medicine. So we did X-ray fluorescence on the tablets, which is a non-destructive technique that can detect the inorganic components that are also present in the tablets, possibly as a colorant and things of that. Using these different techniques and the different devices in the DPAL program, we're able to potentially look at trends that we see with manufacturers. So if a manufacturer is consistently having quality issues, we're able to report that to the respective regulatory agency in the country, and then also hopefully do a faster analysis so that these pharmaceuticals aren't reaching as many patients and harming them.
Thank you. Any questions?
So what have you noticed anything?
If you can wait for the microphone and state your affiliation, name and affiliation, please.
Thank you. I'm Bob Service with Science. And I was just curious if you've seen any trends already in some of these reports back that are coming, and can you describe the network of universities that's being set up to monitor?
Okay, so let me answer those sort of in order. So I'd say the drugs we've analyzed to the greatest extent have been beta-lactam antibiotics, and there we see overall between 10 and 30% of the products we analyze are substandard. And some of them are very poor quality indeed. So we found some samples of amoxicillin clavulanic acid For 14 packages, we found samples in there where there was no detectable clavulanic acid. We have started to accumulate enough data that we can start to say things that certain brands seem to be associated with substandard products and other brands seem to be associated with good quality products. In order to make that statement, you need to analyze lots of samples, so a minimum of 30 to 35 samples of a given brand is what I like to get. And that is a real burden on our HPLC analysis facilities. And so it's been really good to have other colleges and universities step in to take up some of the slack in that assay. So we have partners at University of San Diego, at Hamline, and at Skidmore who have done assays, and there are other universities and colleges like Niagara that are early in the stage of demonstrating system suitability. So these are forensic samples, and so the analyses are suitable for undergraduate students, but they really have to put a lot of care into recording their data and keeping track of the samples. Even things like who's handled each sample has to be tracked. And that, I think, is a useful component of the program because it lets the students analyze samples that matter in a way that's a better mimic of what they'd be doing in an analytical chemistry job. So, more realistic.
Where are the samples coming from?
Samples are collected in Western Kenya.
We have
primarily.
also some samples now from a collaborator at St. Mary's College who's bringing samples from Nepal. So there's many, many locations where the capacity to do what's called post-market analysis is not very high. So, for example, in Malawi, they would really like to analyze medications, but the government doesn't have a pharmaceutical analysis laboratory. So we're hoping to partner with faculty at University of Malawi to do sample collection and analysis. And that's kind of the entry point. Once we start that collaboration, then we can work with those folks on increasing their in-country capacity to do the post-market collection and testing, and also to do their own confirmatory analysis. So.
Further questions? Doug.
Hi. Doug Dollamore, American Chemical Society. I wanted to get just a sense of how big this problem is. I think that is it 1 in 10, 1 in 2 in 10? How many of these? How prevalent are these falsified drugs in these countries?
So of the 157 beta-lactam antibiotics we analyzed, 57 were substandard. So that's a bit more than a third for those samples.
Okay. And I'm going to jump to the end of the press release. It talks a little bit about the possibility of first responders using this technology or some version of this technology to identify drugs of abuse. When you're a first responder and you're trying to help someone,
Yeah.
you can't send that off to a lab. I'm wondering how this technology would work in that situation. Would the first responders have a sample card that they compare to the card that they actually have scratched the drug of a question onto?
Yeah, so we've started collaborating with a forensic lab in Michigan because, of course, you can't get samples of meth or crack or whatever in a regular academic lab. But what we found is that many of the chemical tests that are on this card, so for example, this nice blue reagent on the card is used to detect narcotic drugs in forensic labs, and so we found that the test card picks up many drugs of abuse. We're working actually with a company to try to commercialize that test, and we think it's going to be most useful with a cell phone app that's used to take a picture of the card, and then the test results show up as sort of a color barcode, and computers are good at reading barcodes. And so that's the direction that I think would be most useful for that kind of field use. You know, the person, a first responder in that situation, their main concern would be the patient, not trying to interpret some color test. So.
Further questions?
Thanks. Rich Whitworth from the Analytical Scientist. What's the sort of rate of false positives and false negatives of the paper-based device? Just want to get a handle of,
Yeah.
obviously, you know, you can do confirmatory analysis with HPLC, but
Yeah.
that's not available in some other countries where it might be most useful. So I wondered, you know, how accurate is your test?
Right.
essentially?
For samples that we do in the laboratory, the false positive rate is low, but for field samples, it's running about 10%. And the reason for that is that the test card picks up excipients as well as active ingredients. And in the developing world, there's an enormous number of brands of drugs, and the manufacturers of those brands don't have to say what all is in their product. They just need to list the active ingredient. And so sometimes, for example, when we started analyzing ciprofloxacin, about a third of the samples were showing up positive for starch. And starch can be used as a filler in low-quality products, but it can also be used as a pressing aid, an approved excipient. And so we had to go to the Kenyan Pharmacy and Poisons Board and get information from them, because although the manufacturers don't have to put their ingredients on the label, they do need to disclose to the country drug regulatory authority when they register the drug. So we were able to untangle that, but we still will often run into weird excipients in dosage form. So.
Further questions?
Has this led to any interdictions or stopping of any of the products that?
Yeah. So the product that had the missing clavulanic acid, they put that on a quarantine in all 47 counties in Kenya, and I haven't heard the results of the discussions they've been having with the manufacturer, so I don't know. That case probably was not a case of a falsified product. It was probably a case of a manufacturing quality issue. And so it's to be hoped that by drawing attention to the problem, the manufacturer can take steps to remedy it, and then thereafter they can make good quality products. So sometimes a law enforcement approach is not the most productive outcome. We want the end point for this project is good quality drugs available in the markets where the patients are buying them.
Are you being supported by manufacturers to promote this?
We've received some assistance from big pharmaceutical companies, but most of the products that are purchased in Kenya are not coming from big pharmaceutical companies, at least not the big American or European companies. So, and that's part of the problem, is that there are so many products that are registered for sale in developing countries. It's very difficult for those countries to monitor the quality of those products in the markets. So, for example, in Pakistan, there's more than 60,000 products that are registered for sale, and it's very difficult, to keep track of that many products.
Hi, Doug Dollimore again. I just wanted to clarify just one thing. What is the difference between a counterfeit drug and a falsified drug, as you refer to it?
A counterfeit drug is one where the
The copyright infringement has occurred, so it's like a mimicking of packaging.
It's a mimicking of packaging. So a counterfeit drug could be a drug of that actually contains the correct active ingredient in the correct amount. A falsified drug is one where the contents of the package don't match what's written on the package. So if it says it contains 100 milligrams of amoxicillin and it really contains 75 milligrams of amoxicillin or 50 milligrams of erythromycin, that's a falsified drug.
So we should
Okay.
be reporting this as a falsified drug. and not a
Yes.
counterfeit
Correct.
drug.
Yes, because these paper cards don't detect authenticity. We can't detect which manufacturer produced the product. All we can detect is whether is what the chemical contents are, but sometimes that's enough.
Well, that's the true quality test, right? Rather than you don't. I guess you don't care.
Can we stop you for one moment and get the microphone?
Yeah.
So I was just saying it's very much based on the patient rather than the pharmaceutical companies. They should be monitoring their own sort of counterfeit problems, right? You're really
Yeah.
focused on, is this drug going to do the job? And
Yeah.
frankly, it doesn't really matter where the drug comes from, whether it's
Yeah.
counterfeit or not, assuming it's got the right levels. You mentioned malaria at the beginning, but you didn't. You seem to focus a lot on the antibiotic side of things.
Yeah.
How far are you with because obviously malaria is
Yeah.
a really big issue. Not enough people getting the right drugs, and then when they do get the right drugs, then maybe not the right drugs. So
Yeah.
how far on are you
Yeah.
with the malaria story?
So with malaria, we can actually detect the difference between these pills, but we still, I think we still have a ways to go in developing the test for the active ingredients. So we're detecting right now. We're detecting the excipients that are in the tablets, and that's not as robust a test as I like. So the positive test is weak, right? Because we're not detecting the active ingredient. So right now we have a test, but it takes 10 to 15 minutes to run, and the other tests on the card take 3 minutes to run. And so we need to get the time down.
Right. What are the
So-
challenges to doing that? I've got quite a loud voice. Don't worry.
Yeah.
I can
need to stream it online.
Oh, okay. Sorry.
Yeah.
Yeah. What are the sort of challenges to
To
getting the active
detecting.
ingredient.
Okay, so this drug contains, coartem contains artemether and lumefantrine. And in artemether, the main functional group that we're trying to attack is an endoperoxide. Normally, endoperoxides are quite reactive. This endoperoxide is a brick. And so we've been, running through endoperoxide chemistry. The thing that works best so far is to react it with hemin, which is the heme that's found in blood cells. And that's believed to be involved in the mechanism of action of coartem. And yeah.
So you've got some tough chemistry challenges, right?
Yeah, there are some tough chemistry challenges. So there are many drugs where the functional groups in the drugs are not accessible for color tests that we currently have on the card. So we're really interested in mining the chemical literature. So before 1950, if you wanted to determine what the structure of a molecule was, you would not have a spectrophotometer in your lab. And instead, you'd be doing degradation reactions and color tests. And so there's a very rich literature of color tests that we're looking at. And there's some reactions which are modern reactions, which may also be useful as color tests. And we're really interested in sort of jumping over, from modern reactions and then trying to take them back and use them as color tests. Yeah.
And some of the issues with the color test is actually the reagents that they use. Like a lot of the forensics tests that they use actually involve like concentrated acids, which you can't put on paper. So we have issues with that as well for transferring to paper.
So Bob Services Science again. Can you describe how the paper test works? And is each lane, I assume it's different for each lane?
Yes, so each lane contains different reagents, and in many cases, some of the reagents aren't visible. So for example, lane K is a test for the phenol functional group, and this lane contains precursors that will generate nitrous acid. So there's nitrite ion and there's an acid that's stable on the paper. And then there's an aniline, a nitroaniline, and when that encounters the nitrous acid, it becomes diazotized. We form a diazonium dye, basically a salt. And then that reacts, if there's a phenol, the phenol will react with some base and become deprotonated, and then react with that diazo compound and form a darkly colored diazo dye. And that's what we're detecting. So that lane contains 4 different reagents that are all mixed when the water runs up the lane. So we're forming the reagent in situ. And then we can generate different colors. So different phenols generate different colored diazo compounds. So we can distinguish a compound like acetaminophen from a compound like amoxicillin. So the lanes are not binary tests.
Thanks. Rich Whitworth again from The Analytical Scientist. You mentioned also an app which seems like the perfect partner for this kind of test, particularly if, as you say, it's not a binary test.
Yeah.
Who are you developing that in-house, or are you working with partners on that? Because obviously that's a different, the chemistry is one problem, right? But the device to read the results is a completely different challenge.
Yeah.
So are you working with different partners on that, and how far are?
Yes, I'm working with partners at Notre Dame to develop a neural net-based analysis tool, and that's primarily with Professor Chris Sweet in our Campus Research Computing Center. There's also a startup company in New York has developed their own app for reading the card. And so this is a little startup called VeriPad, and they are trying to commercialize this technology and moving up through sort of business plan competitions. Yes, I'm working with them. It was a really interesting story because the students who were involved in this started out they were working on a senior design project, and they contacted me about using these cards to analyze heart medications. And so not only did they get a senior design project out of it, they started a company. And they've won several business plan competitions. They've won tens of thousands of dollars, and enough to start their company. And one of the students came with me to Kenya in June, actually to... Yeah, VeriPad. That's the company.
Great. Well, on that note, I'm going to thank both Dr. Lieberman and Dr. Bliese. Thank you for tuning in this morning. The archived version of this session will be posted at bit.ly/ACS Live Philadelphia. Please join us for our next press conference, which is today at 9:30, on watching how thoughts and addiction form in the brain. Thank you.